Hopefully this is a little refresher for you from anatomy, where when we talk about skeletal muscle fibers, they have certain types of banding, and then this is due to their striations. And so each fiber has densely packed subunits that are called myofibrils, and these are going to run the entire length of the muscle fiber. So when we look at this picture here on the left, here we have an example of a myofibril, and it's going to run the entire length of that muscle. Okay, and so these are going to be stacked in register so that the light and dark bands are going to align. And if you remember, the dark bands are due to the myosin and the light bands are due to actin. And so those are going to be synonyms with the thick myofilaments and thin myofilaments. Again, thick for myosin, thin for actin. So each of the myofibrils then are also going to be divided into different subunits. And so the subunit is going to be called the sarcomere, and that is from Z disc to Z disc, okay? And so what we see here with the dark banding, again, that's going to be mostly due to myosin, and then here what we have is the light band, and so that's mostly due to actin. So the I band for light band contains only the thin filaments of actin as the primary myofilament, okay? So if we look in this image down here now, here's our I band, and so here this purple is denoting then the actin. All right, so then we have the A band here, and so the A band is going to have the overlap of both actin and myosin. Okay, and so it's going to be the entire duration. It's not going to be split into two sections, but rather it's just termed into one. All right, and then so what we have too is the H band of the sarcomere, and so the H band is going to be the center of where the A band is, where there is no thin filaments. So what we're seeing here then is going to be predominantly myosin. Okay, and then lastly what we have are the Z discs, which again are going to denote the length of a sarcomere. And so these are going to be found right in the middle of the I band, so found within actin. So actin is going to attach to the Z discs, whereas myosin is attached at this H band. So one other important protein that's found within the sarcomere is called titin. All right, so here what we have is titin filament. And so this is going to be a protein that runs from the Z disc here. Let me do this, hold on. Z disc to the M line. Okay, so here we see it all the way. Okay, and so here's one and then here's the other. Okay, and so the function of titin then is to help with elastic recoil of the sarcomere, so in essence of our muscles. So what we have then is that the M line, again in the middle of where myosin is attached and found within the H band, is going to be the center of each A band, and it helps to hold down the thick filaments. So when we look at a myofibril then, what we have is this hexagonal shape to it, and here what I'm highlighting is actually an electron micrograph of a muscle. And so here, this hexagonal shape is going to be the myofibril, and then this SR denotes the Sarcoplasmic Reticulum, and then these M's represent lots of mitochondria because, again, we need lots of energy for our skeletal muscles. The sliding filament theory now is going to be the explanation for muscle contraction. And so let's look at what happens with each sarcomere. So when we have contraction though, the myofiber and all of the myofibrils are going to shorten by movement of the insertion toward the origin of the muscle. Okay, so shortening of the myofibrils is going to be caused by shortening of the sarcomeres, between Z disc to Z disc, okay? So here, and number 1, this denotes at rest, okay? And so at 2 and 3 here in this image, what's happening is that we're getting more and more contraction. So what you see is that the Z discs are actually shortening in length, okay? And so those lines are going to be reduced. So the shortening of the sarcomeres is going to be due to sliding of the myofilaments. And so the length of each filament is going to stay the same though. So here we have, in this picture again, here we have our actin in this purple, and then myosin is going to be that green. And so if you notice, the length of each of those myofilaments does not change. It's just that they're sliding over one another, and that's why the actin and myosin doesn't line up perfectly where they would touch, because then those proteins would actually break. So this allows for that sliding over so that we can get maximal contraction of our muscles. Okay, so then what we have is that the sliding of the filaments is produced by this power stroke of myosin cross bridges, and we'll talk about this in a little bit, that's going to actually pull the actin over the myosin. So again, here we have myosin, and now the actin is coming towards that M line. Okay, and so the A bands, which what we have here is A band to A band, these are going to remain the same in length during contraction, but are going to be pulled toward the origin of the muscle. The adjacent A bands then are going to be pulled closer together, as the I bands between them are going to shorten though. Okay, so here now our I bands here, you see, are becoming a lot shorter. So then the H bands shorten during contraction as well. So here in this yellow, you see that is getting smaller and smaller too, as the actin is going to be on the sides of the sarcomere are pulled towards that middle, towards that M line. All right, so a little bit more on the myofilaments, and let's begin by looking at the thick filament. All right, so again, thick filament is going to be produced mostly by myosin, and so with myosin, this has the appearance of having these globular heads, and so really similar to like golf clubs, okay? And so there's 2 different binding sites that are found on these myosin heads. So the first one is the actin binding site, and the second one is the ATP binding site. So just as you can imagine, the actin binding site is going to be where the myosin interacts with actin, and the ATP binding site is where we'll have ATP that is going to be where it will bind and then be important for muscle contraction. Okay. So then the next one for the thin filament, so here we have that, again, mostly composed of the myofilament actin, but there are also 2 different proteins that we find within it. So this first one here represents troponin, and the second filamentous protein is going to be tropomyosin. All right, so let's start with troponin here. And so this is going to be a component of the thin filament that is important for where calcium is going to bind to it, and so it's going to trigger then the production of the actual contraction of the force, okay? Tropomyosin, though, what we have, this again, filamentous protein. So normally under resting conditions, we have tropomyosin that's going to cover the active sites so that the actin and myosin cannot interact, okay? So this would mean if it was continuously touching each other, then our muscles would continuously be in contraction, and so that would be like a state of tetany. Okay, so what happens then is that when we have calcium, that's going to bind to troponin, and so that's going to create these structural changes that occur within the tropomyosin, and then so this tropomyosin then is going to roll away and exposing then that actin binding site to bind to the myosin binding site on the actin molecule. So then actin and myosin can interact with one another, okay? And so once they interact with the help of ATP, and we'll talk about this next, then we'll have the production of force, and then the sarcomeres are going to shorten. All right, so now let's walk through the cross-bridge cycle and then how it continues on. So ATP first binds to myosin and moving it to a high energy state. The ATP is then hydrolyzed into ADP, an inorganic phosphate or PI, by the enzyme ATPase. So the energy that is released during ATP hydrolysis, that's what's responsible for changing the angle of the myosin head into a cocked position. And so then ready to bind to actin if this site is available. ADP and inorganic phosphate remain attached here within the myosin head, and myosin is in this high energy configuration. So then the next thing that happens is that the muscle contraction cycle is triggered by calcium ions binding to the protein complex of troponin. Okay, so then this will expose the active binding site on the actin. As soon as the actin binding site is uncovered, the high energy myosin head bridges the gap, and then that cross bridge is going to be formed between actin and myosin. So once myosin binds to actin, then inorganic phosphate is going to be released and the myosin is going to undergo a conformational change to a lower energy state. As myosin is going to use its energy, this moves through this power stroke. Okay, so here's that power stroke. And that's going to pull the actin toward the M line. All right, so here, if you notice, it's going to pull then that actin that way towards the M line. So when the actin is pulled towards the M line, then the sarcomere is going to shorten and the muscle is going to contract. At the end of the power stroke, the myosin is going to be at a low energy position. So then, continuing on, after the power stroke, then ADP is going to be released, but the cross bridge formed is going to still stay in place between actin and myosin. ATP has to come in to bind to myosin right here, and that's going to move the myosin to its high energy state again, and so that is going to trigger the release of myosin from the actin active site. Okay, so ATP is going to be responsible again for that release of actin and myosin. ATP can attach to myosin, which allows the cross-bridge cycle to start again, and then further muscle contraction can occur. So if there is no ATP, the muscle would remain in a contracted state rather than a relaxed state. And so this is actually what happens when we pass away, and when we have all of our ATP that's being utilized and no more is being produced, then our muscles are highly contracted, and so it's like a state of tetany until there's a point where the body just can't sustain it anymore, and then everything just releases after we pass away. So an important ion for the cross-bridge cycle to occur is calcium. So something needs to regulate the cross-bridge cycling so that the timing of the muscle contractions can be controlled. And so this is due to calcium ions that are released from the sarcoplasmic reticulum that regulate when cross-bridge cycles can occur. So in resting muscles, the myosin binding site on actin is blocked by a filamentous protein, which is the tropomyosin, okay? And so let me highlight that again for you here, tropomyosin being that filamentous protein on actin, okay? And so the tropomyosin, though, is held together by troponin, so here in that structure, which is a complex of proteins where one of them will bind with calcium. Okay, so then calcium binds to troponin, which is going to induce a conformational change that is transmitted to tropomyosin. Okay, so then the tropomyosin is going to shift and expose the myosin binding site so that the cross-bridge cycle can occur. Okay, so here what we have is the myosin binding site being blocked by tropomyosin, and then here in the contracted muscle, then we have myosin heads that are going to bind to the actin molecules. All right, so in part A of this chapter, we covered the neuromuscular junction and we talked about depolarization. And so the excitation contraction coupling now, this is going to be the link that we have that depolarization of the action potential to now calcium release from the sarcoplasmic reticulum. So talking about the nerve and how it communicates with the muscle and how these two interact, okay? So as a reminder, sarcoplasmic reticulum, this is going to be a modified endoplasmic reticulum, and this is where we have a lot of our calcium that's being stored at rest. And then it's also where we'll have our calcium release. All right, so with this, we have the T tubules, and so let me highlight them for you here. So the T tubules are transverse tubules. These are going to be the essential structures for the excitation contraction coupling, okay? So excitation referring to the nerve tissue, and then contraction referring to muscular tissue now, okay? So the T tubules are going to be tube-shaped, and these are going to be invaginations that we find within the sarcolemma, which sarcolemma is just fancy name for plasma membrane specific for skeletal muscle. All right, so these T tubules are going to go through and penetrate the muscle fiber. All right, and so what we have here then are going to be here in this like pinkish color. Okay, so those are the T tubules. So then the action potential in the muscle fiber is going to be conducted in the interior of the muscle cell along the T tubules. And so on either side of these T tubules too, what we have are these terminal cisternae, and these are going to be responsible then for having calcium channels that are going to be voltage sensors, okay? And so these are commonly known then as these Ryanodine receptors that we find within the sarcoplasmic reticulum, and so these are going to have those sensors that will be triggered when there is a charge, and so calcium will be responsible for that charge. Okay, so the voltage centers then within these terminal cisternae are going to have conformational changes in response to the depolarization from the action potential. And so this is what's going to then cause these channels to open and then allowing calcium release within that skeletal muscle. All right, so here's a great summary on the excitation contraction coupling. And so this is just straight out of your book, okay? But let's just go through it. So again, with the nervous system, we have the motor neuron. So that's going to, again, be excitable. And so the neurotransmitter that it releases is acetylcholine, okay? So that's going to cause depolarization and excitation. And so whenever the motor neuron releases acetylcholine, that's always going to be excitatory for the muscle, okay? So then within the sarcolemma, which again is going to be just like the plasma membrane, but for skeletal muscle. So it's going to bind to nicotinic acetylcholine receptors, and so these are always going to be excitable as well. And then that's going to cause sodium to diffuse in. If you remember, sodium is going to be excitatory for a cell. So then that's going to produce action potential within the skeletal muscle. Then within the T tubules or the transverse tubules, we're going to have the action potential continue on in the skeletal muscle, and that's going to open voltage-gated calcium channels. Calcium, again, is going to be excitable for the cell, in this case, skeletal muscle. So then we'll have the sarcoplasmic reticulum, and here we're going to have calcium that's going to be released, and then it's going to open channels, and then it's going to diffuse in the sarcoplasma, and then the myofibrils then are going to bind to troponin, and then that's going to stimulate contraction, because that's what's going to move then that troponin tropomyosin complex out of the way so that myosin and actin can bind together. And then for all of you visual learners, let's go through the same thing just like the table, but just in a visual format so you can see what's going on. All right, so we're going to have the action potential is going to travel down that motor neuron and at the synaptic bouton, right? And then so here, these are going to be little vesicles of neurotransmitter, and so those are going to be filled with acetylcholine. So the acetylcholine then is going to be released, and it's going to go through that synaptic cleft. And then it's going to bind to nicotinic acetylcholine receptors that are found throughout the membrane found on the skeletal muscle, okay? And so that's going to produce end plate potentials, okay? acetylcholine binding to these nicotinic acetylcholine receptors, that's also going to activate then these blue sodium channels to open, and these are going to be voltage gated, so due to that depolarization, okay? And so this is going to cause depolarization, and remember, action potentials, these are going to be all or none events. Positive feedback here is what's occurring, okay? Just like within the neurons. So then what happens is that within the T tubules here in this image here, that depolarization is going to spread down, okay? And it's going to open calcium channels, okay? And so calcium is going to come into the, we're going this way, come into the muscle, okay, and then it's going to bind to the troponin, and the troponin then is going to unbind, or troponin and tropomyosin, sorry, are going to be moved out of the way from actin, again, so that actin and myosin here can interact, and then actin is going to move towards that M line causing contraction. All right, so after we have contraction, obviously we want relaxation of our muscles. So the action potentials are going to stop firing both within the motor neuron as well as within the motor end plate. And then so calcium release channels are going to close within the sarcoplasmic reticulum. And then calcium ATPase pumps are going to be responsible for moving calcium back into the sarcoplasmic reticulum. And so this is going to move against its concentration gradient. So this is going to be active transport, not diffusing, okay, not diffusion. Okay, so then afterwards we're going to have calcium that's going to not be able to bind to troponin, so troponin and tropomyosin are going to be bound again, and so then the tropomyosin, that filamentous protein, is going to once again block then the myosin binding site on the actin molecule, okay? And so this event will start again once we have another action potential that's stimulated by the motor neuron to start the process all over again.